Okay, so the enzyme Rubisco, there are a few problems with this enzyme, and I'm going to talk about them in this video. Now, most of the material covered in this video is not on the AQA level specification. It's not particularly much harder, but it's not on the specification, but I've included it because it gives you a good understanding of photosynthesis, a little bit more depth of understanding, and a bit more depth of understanding into how enzymes work and energy changes in metabolic energy in plants in particular. But of course, all of those lessons are all relevant to animal cells as well. Okay, so Rubisco is this enzyme which you get ribulose bisphosphate, which is a 5-carbon sugar, and it reacts, it's in the dark reactions of photosynthesis, it reacts with carbon dioxide to create 2 3-carbon molecules called 3-phosphoglycerate. Now, phosphoglycerate is not quite a carbohydrate, it's too oxidized, so it needs to be reduced a bit more. So you need to expend some NADPH, which will go to NADP plus, and also some ATP, which will go to ADP, in order to turn it into triose phosphate. Now, triose phosphate is a carbohydrate, and that can be converted to glucose. It's got the right ratio of carbons and hydrogens. It is essentially a carbohydrate. Okay, so that's what happens in the dark reactions of photosynthesis. And we've trapped a carbon atom there from carbon dioxide and turned it into an organic carbon. So that happens 6 times effectively. You've made a glucose molecule. And that's what's happening in the dark reaction. Now, unfortunately, right, first of all, let's say this enzyme is a very slow enzyme. It's got something called a turnover rate number of 3. Now that means it will, if you've got enough substrate to saturate all the active sites of the enzyme, so the substrate's not a limiting problem, then it can catalyze 3 molecules per second, which might sound a lot, but then if you look at and a lot of enzymes are much faster than that. So acetylcholine esterase, I'll tell you what that does in a minute. Esterase, that has got a turnover number of about 20,000 per second. Okay, this is per second of the units. Acetylcholine enzyme, choline esterase, breaks down the neurotransmitter in the synapse, and that is necessary to break that down very quickly, because otherwise the cell at the other end would keep being stimulated all the time. Okay, so just Rubisco is not a very efficient enzyme. Why isn't it efficient? Well, it's to do with evolution. Rubisco obviously evolved early in the Earth's history, before animals were around. Okay, so at that point you had very high concentrations of CO2, much higher than the current levels, 0.0, 0.3% or whatever it is. So that means it didn't need to be too efficient, because the CO2 levels were much higher. And now they're not. They're pretty low. Also, there is another problem with this enzyme, and that is not only will it react with CO2, but unfortunately, what else will fit into the active site there is oxygen. And so we'll see what happens when oxygen fits into the active site. Right, so when oxygen fits into the active site, here we've got ribulose bisphosphate. We've got a 5-carbon sugar, and it reacts with oxygen. Now, that breaks down into 3-phosphoglycerate, which isn't too bad. So that can be recycled back into ribulose bisphosphate. But of course, you will know you need NAD. You do need NADH for that, NADPH, and you do need ATP for that. So that's a bit of a waste. You're just using NADPH and ATP to get back where you started. And then you make this 2-carbon unit here called phosphoglycolate. And what happens to that is it loses the phosphate group here, and then 2 of those, the various other changes, essentially the the 2 carbon units are combined, but you don't get a 4 carbon unit organic molecule, you only get a 3 carbon, right? And you lose one of the carbons as CO2. So if we started off with ribulose bisphosphate with 5 carbons in there, we've only now got 4 trapped in organic molecules, and we've lost the CO2 molecule. And that's pretty bad. It's the exact opposite of what you want to happen in photosynthesis. And if you think really what is the sum total of what's going on there, then is you end up, you're going to use oxygen, you're going to generate CO2. Now, what other process uses oxygen to generate CO2 is obviously respiration. So this sometimes is a bit misleadingly is called photorespiration, but there's not really any useful respiration. You're not getting any useful ATP or NADH out of that at all. It's a complete waste. You use up O2, you generate CO2, and also you have to then you're going to use NADPH up and ATP, because you're going to have to turn this 3-phosphoglycerate back into ribulose bisphosphate, which requires them. So it's all the bad things and none of the good things there for respiration. So it's a big problem for a plant. It obviously doesn't want that to happen. Now, when does this happen a lot? Well, it tends to happen. The enzyme catalyzes the oxygen reaction much faster when the temperature is high, and usually the cutoff point is 28 degrees C. So you're talking about tropical plants. Plants which are going in tropical environments are going to have a problem photosynthesizing because of this photorespiration thing is going to be going on all the time. It's not so much of a problem when it's colder. Now, what can the tropical plants do about it? Well, they've developed a strategy to get around it. And what they do is they concentrate CO2 in the photosynthesizing cells. And if you can increase the concentration of CO2, you're going to get like a sort of competition for the binding, the active site. You're going to make it more difficult for the oxygen to get in there, so you're going to get less of a problem with photorespiration. Now, how do they concentrate the CO2? Well, the way they do it is this, right? If you think about, I'm going to draw 2 cells. I'm just going to draw it as a box. Now, this one is going to be a mesophyll cell. So you may remember in your leaf morphology, you've got your spongy mesophyll cells, and they're the ones in contact with the air. Right, and then you have got your, in most leaves, we call them the palisade cells, but I think in these kind of plants, they're called bundle sheath cells. And that's getting the most light, so that's where you're getting most of the photosynthesis. And what happens is you get a 3-carbon sugar here, which is you get a 3-carbon molecule here, and that reacts with CO2 to form a 4-carbon molecule. That 4-carbon molecule then goes from, so this is in the mesophyll cell, which is, plenty of CO2 here. That goes into the bundle sheath cell, which is away from the air, but that's where the photosynthesis is occurring. And what happens is that 4-carbon molecule will decompose back into a 3-carbon molecule, and you'll get CO2 released there. And then what happens is this 3-carbon molecule will diffuse, because it's going to be right, it will diffuse back into there because it's being used up here. Okay, you can see here we're constantly using up the 3 carbons. So once you regenerate it, it will diffuse back in down the concentration gradient. It will react with the CO2 and go around the cycle again. But there is a little bit of a price to pay for this, because this reaction, when you get the 3 carbon plus the CO2 and you turn that into the 4 carbon unit, that requires ATP. So there's a little bit of expenditure of ATP in order to concentrate the CO2 in in this photosynthetic cell. But that's what happens in tropical plants. And this was this pathway is named after the two scientists who discovered it, and they discovered it a long time ago. I think it was in the 40s. It's called the Hatch-Slack pathway. So just a couple of things about Rubisco there, why it isn't the most efficient enzyme there is.